Okay, now we'll discuss about the contributions of two important scientists. Actually, in latest syllabus, Harish, mute your speaker.
Okay, sir.
As per your syllabus, we are supposed to understand the contributions of Louis Pasteur and Robert Koch. So these are the only two things are there in our syllabus. But we'll discuss something about Anthony van Leeuwenhoek as well as Edward Jenner, because without their contributions, without their work, we cannot lead further. So I'll do in brief about Anthony van Leeuwenhoek and Edward Jenner. Then we'll go in detail about Louis Pasteur as well as Robert Koch. The remaining scientists, you can refer like when you are reading the books, you can refer the other scientists, but in syllabus it is prescribed to study about Louis Pasteur and Robert Koch. So Anthony van Leeuwenhoek, actually, it is believed that he is the first scientist to develop a microscope, but it's not true. There are several scientists who developed microscope before to him, but he is the first person to observe the microorganisms, actually moving objects through the microscope. Small things like RBCs, some algae, protozoans, sperms. He has observed through the microscope, and he has used the word for them. He, because they are moving, he found them to be animals, and he has used the word little animalcules. Animalcules, because they are moving sperm cells, raining water, like pond water, he has collected and kept it under the lenses. He has prepared simple microscope. Actually, lens, we will be sometimes will be using reading lenses. Such kind of reading lenses he has used and observed some of the microorganisms. Some algae he observed, some fungi he observed through his microscope, and even today also there are like his collections, his preparations. He's having some his collections are still today preserved in Royal Society of London, and like he has explained micro outer sketches of microorganism very well, actually, which can be explained only by using the compound microscope. He has prepared only simple microscope, means single lens microscopes. Compound in the sense, if you are using more than one lens, that is called as compound microscope. So he has used simple microscopes, but see, basically he's a person of the doing business with cloths. There's a cloth, sometimes there's a threads comes out. The quality of the thread, quality of the cloth is measured based on the that thread is coming out or thread is opening up, such kind of things. So he used to prepare, like this used to be hobby for him. Anthony van Leeuwenhoek, he used to grind some glasses. He used to grind, and he used to make one piece out of it, and he used to put it in the frames, and that whatever the joint pieces are there, that he used to use for the magnification purpose. uh, See, it is for him. But he did a lot of observations, and he's the first person to observe the microorganisms. In fact, algae, fungi, and some protozoans, all of these things are microorganisms. He has observed those things, and he has reported to Royal Society. That is the initial studies of Anthony van Leeuwenhoek. Then later on, let's come to the Edward Jenner. Edward Jenner, we call him as a father of immunology. Father of immunology. You know, at that time, there's a variola. There is a smallpox was a very big problem. Variola, variola is a virus name, and which causes a smallpox. Smallpox, you must have seen chicken pox, some blisters kind of things will appear on face and on hands and feet and all those things. And it is see this actually chicken pox appears only on the epithelial tissue, only on the top, only external surface only, whereas the smallpox appears even inner sides also, in the lungs, in the intestine, all like inner coating as well as outer coating, both epithelial tissues gets your smallpox. And see, it used to be believed that once a person gets a smallpox, he has to die. Such a dangerous disease, the smallpox. And even it is a contagious, it spreads from one person to another person. And there's a, in olden days the people used to use smallpox virus, variola virus, as a tool for bioterrorism. And Edward Jenner, see, I'll narrate a small story. What was happened during his time. Variola, actually, it's a related virus like cowpox. That is also variola, but see, the species are different. Mycobacterium tuberculosis causes the TB. Mycobacterium bovis causes it is there in what you call your buffaloes, but it does not cause tuberculosis. So these particular variola species of cowpox, it does not create the disease in human beings, and it causes the some like pustules, some bristles kind of. Sometimes when you got a fever, you will get pustules over here. It will be filled with water and little amount of uh, water and pus, little amount, and it will be coming out something. So this kind of pustules, he observed, Edward Jenner observed in there's a milk lenders, so whoever collects the milk from the cow, in those people, he found that those peoples are getting this pustules or bristles, small bristles on their hands. And to his surprise, he found that those people, that's milk lenders, they are not getting the smallpox disease. And he thought that these two, like cowpox and smallpox, are sister concern, like your Mycobacterium tuberculosis and Mycobacterium bovis, because by that time binominal nomenclature has not established. At that time we don't know well about binominal nomenclature. So he thought that there is a something relation in between this cowpox and smallpox. Smallpox is very well known, and smallpox, he thought that the blisters which are forming on the teats of the wood or teats of the cow could be due to the cowpox, and this cowpox could be somewhat related with the this smallpox. And as these two are similar, this cowpox and smallpox, as they share some commonality in them, these milk lenders, these people who collect the milk and the sales outside, they are not getting the disease. Which disease? Smallpox. So what he did, like he started collecting that's the blisters, the pus or the water that accumulates in the small pustulates. Actually, scientific term, it is a pustulates. So from those pustulates, he has collected the pustules, and whatever the air is there in that, whatever the water is there, pus is there in that one, he has dried that water and pus. And actually, he has collected from a lady, Sarah Saleha. There is a lady, milk lender actually. She used to collect the milk from the cows, and she used to sell in nearby market. So this Edward Jenner has collected the pustules from the hands of this Sarah Nelema, and he has dried it. See, basically, it is not a smallpox virus. It is a cowpox virus. It does not cause any infection. But still, to make sure that it is not creating any nonsense problems, he has dried that pustules, which consist pus as well as water, and he has started like giving it. First, he has given to a boy, small boy called as James Philip. James Philip is the first boy. To him, actually, he has injected this cowpox virus, or the pustules which were collected from the cowpox. It is actually, they were not growing on the cow teats. They were growing on the a milk lender called as Sarah, and he has collected that pustules, dried it, and made a powdery form and suspended in water and injected to James Philip. He is the first gentleman. It was happened open administration, open vaccination, and this James Philip got protected from that smallpox, and that led into the development of a beautiful virulent vaccine preparation. It is a virulent vaccine preparation. This was the first vaccine ever developed in for mankind, and that's what Edward Jenner was considered to be the father of immunology. Okay, now we'll do the two major scientists. There's Louis Pasteur, on whose name, the great Institute of Pasteur in Paris has been established, and whom we call as a father of microbiology. And 27th December, like it was his birthday. Uh, at that time, he used to involve in almost all the activities. At that time, he used to be considered as a major troubleshooter. Uh, and see, we don't need where and how he born, what is his father, and all those things. For us, the most important thing is what are his contributions. Basically, he's a doctorate. He got PhD in chemistry, and he was appointed. In royal families, they used to appoint even for wine testing. Uh, there are specific persons who will be appointed in wine this, because who requires to be good taste buds. So this Louis Pasteur was appointed as wine tester in royal family at that time, and he was given a free freedom. He's a PhD in chemistry, but he was very fascinated in medicine. He was very fascinated in medicine, and but he has end up his work in microbiology. A lot of microbiology work he did. So several things he has started, and he was the first scientist to use the word microbiology. He was the first scientist, and he's the first scientist to use the word vaccine. And see, Edward Jenner has developed vaccine first, and Louis Pasteur later. But the term vaccine was used. In fact, this Louis Pasteur also developed the vaccine for rabies. We'll see later. So it is Louis Pasteur who has used the word microbiology, vaccine, And this Louis Pasteur has discovered the fermentation process. He used the word fermentation. Before Louis Pasteur, people used to think that it is a chemical phenomena. What is the fermentation actually? Here, this sugar molecule, whether it is a glucose, fructose, or whatever may be, this glucose is interconverted into alcohol and acids, alcohols, and mostly alcohol process. That at that time the most popular is ethanol production process. So beverages production process is concerned. So at that time, so people used to think that this glucose gets intramolecular change, it gets splitted. It is a chemical, basically it is a chemical reaction, they used to believe. And later some scientists, they have reported that it is a yeast is involved in this interconversion process, but still they didn't mention that it is a microbial process or living organism involved in this process. Even though yeast is involved, they used to believe that yeast works as a catalyst. Again, it is a chemical reaction. It is basically Schwann, Theodor Schwann, and he is a chemist person. He used to believe that it is a pure chemical reaction. And this, see, Louis Pasteur, reported that there is an enzyme which is responsible for conversion of glucose to ethanol. And if you kill the yeast, if you heat kill the yeast completely, then the capacity of converting into glucose to ethanol gets lost. The capacity to convert glucose to ethanol is lost. See, later that enzyme was identified as zymase. So, and even during this, it is Louis Pasteur who has reported that microbial fermentation is responsible for the production of beverages or ethanol. It is not a chemical process. And during this particular work, he discovered a process called as contamination. Wines, I told you, he's a taster of wine. Taster of, so he used to taste the wine. If wine quality is lost, if you go France, European countries, they will like how we grow the paddy here, our sugarcane, commercial crops. There, the commercial crop is we can say grapes and wine. So the quality of the wine is disturbed because of the contamination. He found through the microscope, in the microscope lenses, he observed that along with the, yeasts are bigger in size if you see under the microscope, like a ball. And if you see, bacteria are very small. And he found that along with your yeast, there are certain other things which are actually hampering the quality of your wine. They are disturbing the quality of wine, are damaging the quality of wine. And he called this process as a contamination. And he told that you have to remove these contaminants, unnecessary things. And actually, he emphasized the importance of sterilization. He told that sterilization must be done before starting a fermentation process. You should do a sterilization. In fact, he has used a pasteurization process. Here, he has used less than 100 degrees Celsius temperature for inactivating this bacteria, and pasteurization process. Okay, pasteurization we'll see later. And he's the first scientist, don't call it first scientist, he's the scientist who has completely disproved the spontaneous generation theory. We discussed it in detail. So earlier people used to think that the life originates from the non-living organisms or non-living matter. And like many scientists has contributed for biogenesis theory, starting from Reddy. And finally it is Louis Pasteur, by using his gooseneck flask or swan neck flask, through which he has proved that it is the biogenesis theory, right? It is not the spontaneous generation theory. Okay. And one more, you must be aware about D form and L form. Amino acids, sugars, sugars are D and L forms are there. And are levo, you must have seen plus or minus position, this carbohydrates, levo and dextro form.
Destro.
Yeah, what are those things? What are those things? Tartaric acid is there. Tartaric acid, levo and dextro tartaric acids are there. What do you mean by are plus or minus? Sometimes left or right, left and right is also indicated. What is this? It is, see, the optical activity we call them. See, based on the active site and remaining branches, whether they are left side or right side, based on there, they will be getting levo or dextro. Why they are having that much importance? They are having the, see, it is the Louis Pasteur first who discovered these optical activity of macromolecules or organic molecules. Amino acids, you take the amino acids, only L form of amino acids are utilized in our body in any living organisms. D forms, like in peptidoglycan structure, a few D amino acids are involved, whereas D forms nowhere we'll be using. Whereas sugars, only D form sugars, whatever you dextrose or whatever the sugars that we consume that are involved with our living organisms, only D forms, no L forms. L forms are not there. L forms are there, but they are not active. They are not involved. They are not involved. So this optical activities of organic compounds was reported first time by the Louis Pasteur. And he is the first person to cultivate microorganism in liquid media. He is the first person to cultivate the microorganisms in liquid media, and he has proposed that before cultivation, like see, fruit pulp is a liquid media for him. Fruit pulp, whatever the grape pulp was liquid media to him. So he has advised that first of all you should do sterilization of whatever the media that before cultivating the microorganisms. That is being practiced nowadays. Sterilization of, he has insisted the need of sterilization. He has insisted the need of sterilization before doing the autoclaving. And he has proposed the sterilization at 120 degrees Celsius. He has proposed the sterilization, the process of sterilization at 120 degrees Celsius. And sometimes if it is a spores are there, or if you are doing the dry heat sterilization, it should be done at 170 degrees Celsius. Now we are doing at 160 degrees Celsius in hot air oven, and 121 degrees Celsius we are doing in autoclave. Okay, and there is one more contribution of Louis Pasteur. You know, Reddy's experiment. What he did, he took a loaf of meat and he kept it open in one, another one sealed, and the third one covered with a cloth. Isn't it? And he was found, Reddy was found the putrefaction, but he didn't observe that what is the causative agent of it. He didn't found, he didn't explain why that putrefaction, there is a damage of meat loaf or boiled meat is taking place. And it is Louis Pasteur who has reported that microorganisms are responsible for this putrefaction process. This putrefaction of meat loaf is due to the microorganisms. And later on, like for milk sterilization process and wine sterilization process, he has introduced a concept called as pasteurization. Pasteurization is less than 100 degrees Celsius. Generally, we'll use 55 to 60 degrees Celsius to reduce the microbial load, not completely. See, pasteurization, objective of the pasteurization is not to make zero microorganisms. Sterilization objective is to make the microorganisms zero, whereas pasteurization objective is to reduce the microbial load. If the microorganisms from 1 million, if they can reduce to 10 or 100, So it takes time to multiply for them, so that we'll be having a lifespan of milk for a day or two. So it is, two things are different. Pasteurization is not sterilization. Pasteurization, our activity, our job is to reduce the microbial load, not to make it zero. So it is Louis Pasteur who has introduced the concept. Why he has done pasteurization, not sterilization to the wine and milk also? Why do we do pasteurization of milk? Why not sterilization? Anybody? Amarnath?
Sir, because it is a microbial mediated activity, that's why if we do sterilization, the microbe which is helping in the conversion of carbohydrates to alcohol will also be killed.
No. Amarnath?
Sir, please repeat, sir, once again.
So the milk and wine, he didn't do the sterilization process. Sterilization is generally we do a 121 degrees Celsius, 120 degrees Celsius, and but he has gone for 65, like 55 to 60 degrees Celsius. That process is called as pasteurization. Even milk also. Milk we are not doing like 121 degrees Celsius. We are just boiling it around 65 degrees Celsius. Why? Actually, I have given the answer, but I want more scientific explanation.
Proteins in the milk will damage.
Yeah, actually, denaturation of proteins takes place if you do there is a heating at above 100 degrees Celsius. That's the one reason, and actually that's why. Okay, one more further. Why not wine? Wine? Why you are not going for boiling it above 100 degrees Celsius? Why you are doing 55? Wine 55 will be using. Why? Because the alcohol content. See, first I will complete this Gautam's answer. See, he has mentioned that. Gautam, can you repeat once again?
Yes, sir.
Yeah, please tell me your answer once again.
The microbe which is involved in the conversion of carbohydrates to alcohol will be killed, sir, when we heat above 100 degrees.
See, your wine preparation, once the wine preparation process is over, there is no need of your microorganisms. You can kill them. Absolutely, there is no problem at all. Actually, all microbial products, they are killed. Microorganisms are killed after the process is completed. So there is no absolutely problem of killing the microorganisms. Okay. And here, if you do above 100 degrees Celsius, whatever the ethanol is there, that gets evaporated. You will not wine without alcohol. Nobody will accept it. That is why you are not supposed to do above 100 degrees Celsius. So it is pasteurization is recommended. And the microbial load is concerned. See here, somebody, you must be having a doubt, sir. Still at 55 degrees Celsius, microbes are surviving. Microbes are surviving, but their number got reduced from 1 million per ml to it has come to 10 microbes per ml or like that one. So if the number gets reduced, it takes time to substantially their growth. Got the point? And in the milk, as Deepak has mentioned, see, the main reason of the protein milk gets denatured if you are doing it at very high temperature. So if the protein gets denatured, that is non-digestible. And you must have seen small babies, or even sometimes you people also, if you are consuming overheated milk, charred milk, otherwise you can call it as, if you are consuming it, that is not digested, and the kids will do the vomitings. Okay, so this is the importance of pasteurization process is concerned. And it is in 1865 he discovered another disease called as, he didn't discover actually, at that time I told you whatever the problem is there, he used to be a troubleshooter. So febrin, in silkworm there was a disease, even today that is there, febrin. It is a protozoan disease. It is a protozoan disease, and the silkworm industry people, farmers, approached Louis Pasteur to give a solution to their problem. He observed the cocoons. Cocoons are, you know, that silkworm fruiting bodies. So he observed those things, and healthy versus normal, and observed under the microscope also. And he observed that it is larger than that of bacteria. The causative agent is larger than that. It could not be. It should not. He said to himself. He said that it should not be a bacteria, and it is doing holozoic nutrition. It is taking up pincocytic and holozoic nutrition. It is doing, so it should not be your bacteria. So pasteurization or heating process will not support in this one. So it is then he has reported it as a bacterial one, febrile disease, and sorry, it is a protozoan. And he reported that to use disease free fresh cocoons. He has proposed, he advised the farmers to collect the cocoons or the starting seed material, which is a disease free. And actually, this was a very good contribution to the silkworm industry. Then, fowl cholera. He developed the first vaccine for the fowl cholera, poultry cholera. What is the causative agent of poultry cholera? What is the organism causes cholera? Vibrio cholera for human being. What about fowls? This is Pasturella maltosida. Pasturella maltosida causes fowl cholera, and what he was doing. He was cultivating. He was cultivating that organism. He has isolated. It is a Pasturella cholera. It is a bacteria or fungi or virus. Pasturella maltosida. You have studied in intermediate. It is a bacteria. Okay. He has isolated that bacteria, and he was growing that bacteria and kept it in the fridge for some time. He's left for some time, and he came back, and when he observed under the microscope, actually this Pasturella maltosida, Vibrio cholera, both the things, they are having the flagella. They are having flagella. So this flagella, if you incubate the bacteria in abnormal conditions, like in fridge 4 degrees Celsius, for a certain time, it will lose its flagella. And what he found is he has injected that the Pasturella maltosida, the cholera causative bacteria, which was incubated in the fridge for a week period, to the chicks. That is chicken chicks. And he found that, to his surprise, he found that there is no cholera disease. You're getting my point. He was growing the cholera bacteria, fowl. There's a chicken cholera bacteria, and when the newly isolated bacteria, if you are injecting into the chicks, they develop cholera and they will be killed. But this bacteria, if he has incubated for some time in the fridge at 4 degrees Celsius, that is abnormal conditions. Generally, this bacteria will grow at 37 degrees Celsius. So at abnormal conditions, the bacteria which was grown for some time in abnormal conditions was found to lose its flagella. This is one external observation that he found. And when he injected that, there's a flagella lost bacteria, what he found is the chicks didn't get any cholera, but subsequently when you give, see, this is given by nasal spray. When you give that active culture also, in these particular chicks, they are not getting cholera. Means this is working as a vaccine. This is working as a vaccine. He has developed a vaccine by eliminating its potency. The ability of causing the disease of fowl cholera or chick cholera has been lost by incubating it in unfavorable conditions. Okay, this is called as evaluation, and this is being used for nowadays for vaccine development. Okay, and next, anthrax. Anthrax bacteria was isolated by Robert Koch. A lot of work was done by this Robert Koch, but it is Louis Pasteur also who worked on the anthrax. See, these are the contemporary scientists. These two are the contemporary scientists, and that era, 80, there's a 1850s to 75 is actually golden era of microbiology. These two people worked a lot during that time. Okay, so he worked this anthrax bacilli was also grown both the scientists, and see the causative agent as anthrax was first reported by the Louis Pasteur, and Louis Pasteur has successfully cultured staphylococcus, which creates the throat infections to us, and it is a common in our skin. It is a common it is there, and he's also isolated. There's a this he has inoculated this staphylococcus aureus in rabbit, and he has grown it in the animal models also. So he's the first person to to use the animal model. And the major contribution is concerned of Louis Pasteur. He has developed a vaccine for rape. Okay, rabies. Rabies is a viral disease, and the rabies virus is in bullet shaped, and the disease also called as hydrophobia. Basically, this rabies virus acts on there's a respiratory muscles, especially this bronchiolar and bronchiolar, as well as what is that? There's a neck muscles. So complete stretching of these muscles takes place, and the person if he's taking even water also, it gives a lot. It gives a lot of pain. That's what he's scared of, the pain of even water sound also he does not base, and this is the problem of hydrophobia. So what he did, this Louis Pasteur, he has inoculated this rhabdovirus, rabies virus. So he has inoculated into the sheep, and basically it grows in the nerve tissue, it grows in brain as well as spinal cord. It grows, and once this rabies symptoms develops over there, he will be sacrificing that sheep, and he will be collecting brain as well as spinal cord. He will be making a powder of it. He will be drying and making a powder, and he will be treating with, earlier he has incubated low temperature for inactivating of Pasteurella maltosida, that is foul cholera bacteria. But in this case, he has used sunlight as well as alum. You know alum, patika. This he has treated this. There is a brain as well as brain and spinal cord tissue, which is collected from the sheep, and he has treated it with the sunlight as well as and he has given, see, this is also a public vaccination. Joseph Mister is the first boy. He got actually he got a bite of mad dog, and even we used to call that kukakatuku chawdeba something. And he has given a vaccine, his own developed vaccine to Joseph Mister, and he was survived. And this was a very successful incident at that time. And see, in 1887, this Louis Pasteur and along with another scientist, Jobert, they have described Clostridium species, the septicemium, which causes gangrene, septic problems, and Vibrio septica. These two were discovered by them, and because of these so many inventions on his name, the Pasteur's Institute was established in Paris, and he was the director. And when he died, he died in September 28, 1895. He was buried there, and his tomb is also still there in the Pasteur's Institute. This is about Louis Pasteur's contribution. So almost he touched all the fields. So these are the few that I have listed, almost 18 contributions. But apart from this also, there are several contributions which we can come across in his book. Now we'll see Robert Koch. Robert Koch, Louis Pasteur actually, as I told you, these are the contemporary scientists. And in fact, it is Louis Pasteur first, and afterwards, Robert Koch. And inventions of Louis Pasteur has inspired Robert Koch a lot. And whatever the inventions of Louis Pasteur are there, they were materialized into techniques by Robert Koch. That's what this Robert Koch is considered as the father of microbial techniques, as well as he is the father of medical microbiology. Robert Koch is called as father of microbial techniques as well as father of medical microbiology, because his postulates of Robert Koch, which we discussed already, they laid the foundation to the medical microbiology. They laid the foundation to medical microbiology work. That's what it is to be. It is given a lot of importance to him. See, first, it is Robert Koch who has started work on anthrax. Later, cultivation of this anthrax in liquid media was carried out. And see, it is Louis Pasteur said that it is the anthrax bacteria which is responsible for anthracis. But initial work was started by, actually, the main credit of anthrax goes to Robert Koch itself. He did a lot of work on this Robert Koch. He explained about the sporulation process in anthrax. This Robert Koch has explained about the process of sporulation. Endospores formation was explained first by the Robert Koch, and even he has explained how they get germinated. He has grown this. What he did is, vitreous. There's a buffalo's cattle's eyeball. In that one, there will be a liquid which is called as vitreous. In that vitreous, this anthrax bacilli was used to grow, and when you add few bacilli to it, and see this anthrax, it is like your sugarcane, many chain-like structure. It will be there, anthrax, and it will be a thread-like structure. It will be coming. So when you inoculate a little amount into vitreous, animal vitreous, eyeball vitreous, so a lot of bacteria will be growing in a short period of time. Within a short period of time, lot of bacteria will be growing in that one. And once the nutrients are exhausted, like 12 to 16, 18 hours, as the nutrients are exhausted, the bacteria will start sporulating. How you will be knowing the sporulating? The central round and strong structures will form that you can see through the microscope. And again, you take this one or two spores and you put it into the fresh medium, vitreous medium, then again it starts germinating. It again vegetative cells, like spore cells, sporing cells will be disappeared over there. See, these spore cells will be germinated into the vegetative cells. Okay, this is was his observation, and he has grown it in the and see, to prove that Koch postulates, what he did, like our August Wiesmann like experiment. He has injected this anthrax bacilli into the mouse, not mouse, rat, a wild rat at that time, and he found that this anthrax was entering into the lungs, and it is creating the pneumonia and anthracis, and the mice used to be killed. And from there, he's collecting the blood and injecting into the other mice. Like this, he has continued for 20 generations. I mean, even after 20th generation, he found that the anthracis bacteria is still able to cause the disease. Still, it is capable of causing the disease. After this observation, he started doing almost same work. He did with tuberculosis, several organisms he worked, same phenomena he wants to do. So he has isolated the bacteria and grown in the laboratory, and that bacteria injected again into other mouse, and that mouse got the disease, and from that disease he has observed again the bacteria is there, again he's inoculating. Okay, so basically he was very keen about pathogenesis. Is very keen about the pathogenesis. I told you he has used vitreous. There is an eyeball vitreous for cultivation of this bacteria, and his that was first natural complex media actually. And in 1877, he is the first person to prepare the smear and started staining. Smear, that culture will be kept on a slide for the staining purpose, that is called a smear. And that smearing the technique, that's what he's the father of. Not only this, several techniques he has developed. So he has developed a process of smear and staining. So why we should do the staining? Anyhow, that also I'll be dealing. That's staining, microscopy and staining part I will do, and rest of the things remaining teachers will do. So bacteria are very small. They're transparent. They don't have any color by virtue. Even though some euglena is colored, but as it is a very small, transparent, directly without staining, we cannot see them. So generally the bacteria are colored, staining in the sense colored. Okay, that process was first introduced by the Robert Koch. Okay. And it is in 1878 he observed the wound formation process, and he linked it with the bacteria. He observed the wound formation process, tissue damage. After the cut, whenever there is a cut or injury, then the bacteria will enter into it, and the macrophages will come, and the macrophages will colonize over there, and they will try to engulf the bacteria. As the bacteria, if the bacteria dominates over there, then it will be the wound formation will be taking place, and healing will be taking place more time. If the macrophages are more, then healing of the wound will be taking place. This was Robert Koch's observation. Wound healing process was explained by the Robert Koch. And again, another most important observation is there's a pure culturing. For pure culturing, you require solid media. Solid media is required for pure culturing purpose. So solid media, first potato chips, boiled potato chips were used by this Robert Koch. So he has inoculated bacteria, microorganisms, but some microorganisms, they will digest the starch. They will eat away the starch of potatoes. So what happens, the potato slice, whatever is there, that will be digested. So that was not that it solved the purpose, but not at larger extent, only at shorter extent. Then he has on his wife's recommendation, he has used gelatin Gelatin. Who is the girl here? Gelatin. For what purpose we use the gelatin? Are you using the gelatin for only for explosive bombs? For what purpose will be using the gelatin? Girls, somebody answer. You must have used during your Christmas, and you must be using it for New Year for cake preparation. For solidification of cake, we'll be using this gelatin. So the same thing, like Robert Koch's wife advised him to use this gelatin. You know, gelatin gets solidified at 20 degrees Celsius and it gets melted at 30 degrees Celsius. That was the limiting factor. There afterwards, he has discontinued it. Then another, like his scientist's wife, his student's wife she recommended to use this agar-agar, and he has used the agar-agar for solidification, and now still today we are using the agar-agar for the solidification purpose. And by using the solid media, he has purified the culture. So he has created the solid media, he has created the concept of pure cultures. Pure cultures are very much required for the microbiology work. And he is the first person to work on Mycobacterium tuberculosis. He has identified the Mycobacterium tuberculosis. He developed the staining process of Mycobacterium tuberculosis. He developed the process of in vivo detection of Mycobacterium tuberculosis. Very significant. And even, see, you must have seen for whenever you are having when you are suspect TB, some tuberculin test is done. Small level of injection is given to you here. And if any allergic responses are there, that indicates you are having TB. Actually, he thought it to be used for treatment. Robert Koch thought it to be a treatment, but now it is a diagnostic process. So it is a membrane protein. This is a Mycobacterium tuberculosis membrane protein, which was extracted, and that is given as an injection. And suppose already if you are having antibodies, if suppose a person is infected with Mycobacterium tuberculosis, then definitely he will be having the antibodies to the membrane proteins. So whenever you give an injection of tuberculin, then it will give that will react with antibodies, and it will give a reaction over there. So this is he has developed, he has cultivated the Mycobacterium tuberculosis. And still today the cultivation of Mycobacterium is very tough job, because it's a fastidious. It requires lot of nutrients, vitamins. So he has developed media, and he developed the staining process. He developed in situ, in vivo, like differential staining of tissue, as well as this Mycobacterium tuberculosis was discovered by the Robert Koch. And he has also discovered the Vibrio cholera and Egyptian ophthalmia. These two bacteria were also discovered by Robert Koch. And he explained Koch postulates, which we have explained already. I will explain you again. Tuberculin for diagnosis of Mycobacterium tuberculosis. And he observed microorganisms in water, soil, and air. He worked the microbes of wire. And he developed a hot air oven for steam sterilization process. Autoclave by Louis Pasteur. Hot air oven by Robert Koch. See, it is a dry heat sterilization process. Hot air oven, where it's an oven kind of thing. You must be using the micro oven at your home. Similar thing. Deepak, I know. Let me complete my time. This is the first and last time warning. In my class, if you are disturbing, maybe you must be having the chemistry class. That fellow will wait.
Okay, sir.
Okay. I know my time. I will come always planned. I'm not going vague. I will go as per my schedule. No need to comment on my what you have after and what you things. Okay. So as he did a lot of work on this microbial diseases, anthrax, for cattle disease, and he worked on Vibrio cholera. He worked on Mycobacterium tuberculosis, and based on his observation, he has made some postulations or laws or proposals. Laws which are called as Koch postulates or Koch laws. There are four, as we discussed in the earlier class. They are in a one particular disease, its causative agent must be there always. Suppose if it is anthracis, always anthrax causative agent must be there. If it is a typhoid, typhoid causative salmonella typhi or paratyphi must be there always. Okay, that is the first law. Second one, the causative agent can be isolated and grown in the laboratory. It can be isolated and grown in the laboratory. So what he used to do, he used to take out the blood from the diseased organism and used to grow in the laboratory. Third point, these the cultured disease causing microorganisms, when injected into the host, fresh host, it is having a capacity to cause a disease. That is the third point. And fourth point, again, the fresh organism will develop a disease and it will be harboring the microorganism of disease causing capacity. But there are certain limitations, very few limitations of this. See, there are golden standards till today for the medical microbiology work. For the medical microbiology work, still there are the golden standards. And limitations are concerned, like first postulate says that always an organism should be there in a one particular disease. There are certain diseases like nephritis or pneumonia. Pneumonia, you have viral pneumonia, different viruses, influenza, adenovirus, different viruses, and bacteria, different bacteria are there, diplococci, streptococci. So different kind of conditions are there, but same again pneumonia. But now it is differentiated like bacterial. It is, we should not call as a influenza pneumonia. We should call adeno-pneumonia as per the latest terms. We should actually, that was the limitation. And second point, the bacteria should be artificially synthesized in the laboratory. So there are certain conditions where you cannot grow the bacteria in the in vitro conditions, in laboratory conditions, like chlamydia, rickettsia, you cannot grow. Viruses also can be grown in cell lines, that is a laboratory conditions, but chlamydia and rickettsia was not able to grow, but now we are growing that one also. And another point, when you inject the grown bacteria, when you the grown bacteria into the host, it should establish the disease. But when you keep it in unfavorable conditions, like the fall cholera vaccine developed by the Louis Pasteur, what happened, if you preserve the bacteria in unfavorable conditions for a longer while, it may lose its capacity to cause a disease. There is a virulence, it is called as. So in such cases, you may not be able to get the disease. So these are few drawbacks actually, which were explained later, which were very well explained. We have a solution for it. So still today, the Koch postulates are the golden standard protocols for microbiology in general, and medical microbiology in specific. This is about the contributions of scientists is concerned. Next class we'll do the microscopy, and afternoon we are having a class. Probably Anju madam will be taking whatever the topic that she is covering.